Hi everyone, it's Mr. Gann again. Um, this time I'm going to be talking to you about recombinant DNA technology. I apologize in advance for the way my voice sounds. I haven't been feeling very well the last couple days, but I wanted to get you out these video lectures, so I apologize if I sound funny or if I'm coughing through this presentation. You kind of just have to bear with me. So yeah, recombinant DNA technology. Essentially, what recombinant DNA technology is, if you haven't already learned about it from either your bio teacher or your classmates in their biotechnology projects, is when you take two separate DNA molecules and you essentially put them together to form one singular recombinant DNA molecule. It's used in a lot of things. It's, to be honest, one of the backbones of a lot of those genetic technologies that you had talked about in your biotechnology presentations, hopefully. And so the goal of this presentation is kind of to go through the actual processes, the actual methods, and the kind of the, the tools that are used to create a recombinant DNA molecule. So this is the outline of the presentation. I'm actually going to go through in a little more detail the actual process, like I said, starting from isolation of the DNAs of both the donor and the vector DNA, followed by the fragmentation or cutting of the DNAs, going into the joining of DNAs, the transformation, and then the final slide, I'll talk a little bit about the uses of DNA technology. I'm going to put a little extra focus on genetically modified organisms, or GMOs, and then some of the ethical problems that arise with things like GMOs. Alright, so the first step is a pretty simple step, is just the isolation of our DNAs. So like I said, our DNA is made of, is going to be made of two different DNAs, right? We have something called our donor DNA and we have our vector DNA, right? Each of these is going to be used to create one singular recombinant DNA molecule. Our donor DNA is in many cases the actual particular gene or trait that you're actually trying to replicate or duplicate or to place in another DNA. And our vector DNA is simply our carrier. So that's like the host that's really going to take in the small section of the donor DNA in order for it to be duplicated and replicated in further succession. We'll talk about that later. But generally, the most common vectors are bacterial plasmids, or they're like circular pieces of DNA, so they're in a circle, or macrophages. Enzymes are used in order to isolate a particular DNA and the vector DNA. When you're trying to isolate, animal DNA, you're going to use something called lysozymes, followed by cellulase. This should be an A, not an O, in plants, and then something called chitinase in fungus. Alright, so the next step is the fragmentation of DNA. And when I mean fragmentation, I actually mean the cutting of DNA. So after DNA is isolated, both the donor and vector DNA, in order to be cut, they're treated by something we call restriction endonucleases. Essentially what those are just enzymes, so we call them restriction enzymes. Restriction enzymes. And those are the actual things that are going to cut the DNA into fragments. So each, as you can see in this picture below, we have a plasmid from a bacteria and then our DNA of interest. And eventually, as these arrows show, they get cut, right? And they cut into different fragments. As you can imagine, those restriction enzymes have a very particular place on each DNA that it cuts at. It's a specific site, a specific sequence, and we call that the restriction site. And all created restriction endonucleases, excuse me, are going to cut at a palindrome sequence. If you don't know what a palindrome sequence is, or a palindrome is like a word or a series of letters that are read the same forward and backwards. Like an example of a palindrome word is the word race car, RACECAR. It's read backwards, read forwards and backwards the same way. So one particular restriction enzyme is called EcoR1. It's a pretty commonly used restriction enzyme. It's made from the bacteria E. coli, and it has a particular palindrome sequence of what it cuts. And when it cuts it, it creates something we call sticky ends. And this is this little section right here over in this drawing where it kind of looks like it's a little piece that is jutting out. We call those sticky ends. And I'll draw you a little diagram at what I'm talking about. So imagine we have a DNA molecule, right, 5' and 3', in an anti-parallel matter, and we have two complementary strands, GA, T, T, C, C, T, T, A, G. So 5', and we are in our anti-parallel matter. As you can see in here, we have a palindrome sequence. So it's cutting here and here, right? So this little segment right here is our palindrome sequence, and this is going to be that indicator, and this is that restriction site that EcoR1 is going to come in and create and break the bonds in between the DNA. So eventually we'll have a piece that looks like this, 5', 3'. So one in the DNA is going to have just a G on its first strand, and this one, T, T, A, excuse me. Our other side would have like this. T, A. Right? So that's what's going to happen. As you can see, we have sticky ends here and here, as well as here and here. So that's what we call the palindrome sequence, and that's what we call sticky end, and that's the real job of the restriction enzymes. It creates those, cuts those, both the donor DNA and the vector DNA into different fragments, which then allows them to be joined together in like a smooth fashion later on. So like I said, the next step is the joining of DNAs. It's essentially the combining of both that donor DNA fragment and the vector DNA fragment, and then annealed with the enzyme ligase. So as I said, like for example in this diagram, we have the human DNA over here, and we have our plasma, or our vector, or carrier. So what's going to happen is that you can see that EcoR1 is the restriction endonuclease that's being used, and it's going to cut at that specific restriction site, right? That sticky end site. It's going to cut those, and it's going to cause a small fragment of the DNA to be like kind of cut out and able to connect, because as you can see, when you cut our plasma, or our vector DNA, because we use the same restriction enzyme to cut both DNAs, now they are complementary to each other. So those sticky ends can match up with the sticky ends of the opposite donor, right? So eventually both of these, because now this space matches up perfectly with this small fragment of our donor DNA, and they can anneal, and after the process of ligase coming in and ligating them together, our small donor DNA fragment can be combined with our vector DNA plasmid to create a single complete recombinant DNA. So this is, when I say recombinant, once again I mean the actual combination of two different DNAs together. Okay, so the last step is called transformation. When I mean transformation, I just mean is when that new DNA recombinant molecule is inserted into a bacteria cell, either through microinjection or a gene gun, something like that. And then the bacteria cell, will then eventually take up that recombinant molecule to duplicate and divide. So as you can see in this diagram here, we have two cells that have been inserted with our new recombinant DNA molecule. Excuse me. And then through a process of replication, amplification, and cell division inside this bacterial cell, that recombinant DNA starts to duplicate and divide inside of the cell, and then eventually um, those cells itself replicate and divide, and only to duplicate even more. Eventually, this is how we end up with entire clones and things like that, entire actual organisms with the same DNA, is because through the process of transformation. All right, so some of the ethical issues. So now that we've talked about the steps and everything, we're going to talk more about the actual uses of recombinant DNA technology and some of the ethical issues behind it. As I said, one of the bigger uses of recombinant DNA technology is producing genetically modified foods. So, obviously GMOs have a lot of ethical issues, um, but they are rising in popularity now, and especially I think it's 75% of all foods that you find at like a Safeway have GMOs in it, and the rising popularity due for multiple reasons. One is that GMOs, if done correctly, can produce large, much higher amount of food. That higher yield is what makes them quite popular. And this may end world hunger and things like that if used properly. Also, they have quite popularity because of the benefits and what you could actually do um, with creating GMOs. A lot of people have selected traits like herbicide resistance or pesticide resistance. Um, and, there's all those certain traits that make GMOs quite popular. One case, there's a gene in a mango tree um, that is taken and placed in tobacco to kind of fight off the negative effects of increased salt levels in soil, because mangroves are a coastal plant, so they often have to kind of be able to handle an increase of salt because they're so close to the ocean and saltwater. So someone saw that as a benefit and decided to take that, use that as our host gene, put that into a vector, and then eventually place that back into the cells of a tobacco plant, which then could be propagated through transformation into creating a genetically modified tobacco plant that could handle increased levels of salt. So obviously there's things like that. There could be health benefits. Um, the ability to create different medicines and things like that from GMOs. There are certain potato plants that are being used to um, help fight some health diseases. But along with that, there could be problems with GMOs, right? Unfortunately, it's actually really hard to prevent cross-contamination between GMOs, because if you have an entire field of GMOs, like how can you really stop those from cross-contaminating with a neighboring field of, just regular non-GMOs plants? It's very difficult to do. Plus, unfortunately, we don't know the health effects of GMOs and whether they're actually unhealthy. There's been a large debate that, they actually have a negative effect on health. Um, but it's really hard to tell because one, research is just not finished. It's still an ongoing thing. And also, GMOs are not labeled. There was a law that was proposed to label GMOs in this past couple years. However, it never passed. So we've probably been eating GMOs for a while, and so far we haven't really seen negative effects. But to be honest, it's just too early to tell. So it's very difficult. So, um, yeah, that's basically it. If you think about what cloning is, essentially it's the use of recombinant DNA technology to clone particular traits and to pass it on. Cloning within itself, just like GMOs, have its own ethical issues, which I'm sure most of you can devise um, on your own, either by just kind of thinking about what cloning is, thinking about diversity, um, and things like that. So I'm going to end my lecture here. If you have any questions, feel free to write them in a comment on the YouTube link that Alex gives you. Feel free to come ask me. I'm in either B104 in Mr. Hajarian's room, because that's where I teach, or sometimes I'll either be in Mr. Jones' classroom or in Miss Smith's classroom next door. So feel free to stop by. If you have any questions either on this or about coral bleaching, both I have a real passion for. So don't be afraid to ask, and hope to see you soon. Thanks for listening.